Okay, so let's talk about the last part of the cytoplasm, which is the nucleus. We've talked about these different sections. First, you have the plasma membrane around the outside. It's a major component of a cell. It keeps the inside separate from the outside. What was the name of the fluid inside? Right, intracellular fluid. What's the name of the fluid outside? Extracellular fluid. What are the name of these little structures that have functional purpose in the cell? The organelles, and they sit in the cytoplasm, right? And then you could probably name some of these organelles. Like, what's this one here, the membrane-bound organelle with these zigzags back and forth that make energy? The mitochondria. How about the stuff that comes off of the nucleus that makes, well, what's this rough stuff? This rough endoplasmic reticulum. What's it make? It makes proteins. What's the name of the smooth version of this that sits just off of it? Right, smooth endoplasmic reticulum that helps process or modify carbohydrates and lipids, synthesized lipids and fats. And what's this shipping compartment called? The Golgi apparatus, right? Sometimes called the Golgi complex. What are these little carrying things called? Vesicles. What are these long tubes here called? Microtubules. What are these crisscross things that like to move to poles called? Centrioles. So you have a lot of these structures. You already know what they are. Let's talk about the major structure in the center. So if you're looking at all these structures as important parts in this little biodome, the nucleus is super important because it stores all the blueprints for life. So it stores all the blueprints. Anything you want to make, you need a blueprint to make it. So if you want to make a new piece of rough endoplasmic reticulum, you have to have the DNA. If you want to make a new microtubule, you need to have the DNA. If you want to make new vacuoles or vesicles, anything you want to make that's new, if you want to make a new chemical like insulin, you need, or a new receptor, you need to have the DNA. Cells that don't have DNA they can't live for very long. Like for instance, a red blood cell has lost its nucleus, which means there's no DNA inside of it, which means it can't repair itself. It can't replicate its organelles. In fact, all its organelles are dead and they're gone because there's no nucleus. So the nucleus is super significant. Here's the anatomy you need to get familiar with the nucleus. The nucleus is called the little nut or the kernel. So when they were looking underneath the microscope and they kept noticing there was this little nut or this little thing that looks like a kernel inside of each of the cells, they call it the nucleus. But the nucleus is only in eukaryotic cells. Eukaryotic cells are you. They're human cells. They're animal cells. They're plant cells. Not in prokaryotes. Prokaryotes are things like bacteria. So if you look under a microscope and you see this little tiny cell wiggling around, there's no nucleus inside of it, you can almost assume that it's going to be a prokaryote or a bacterial type cell that's wiggling around. So the nucleus is the control center, is what a lot of people say. Some people refer to it as the brain. I don't really get into that analogy, but if it works for you, that's great. I like to think of it as the library in my little city. Right? The library is the nucleus with all the blueprints, the recipes for everything I need to make in life. If I want to make something, I ship it out. I ship a copy of that recipe or that blueprint out in the form of something called RNA. That RNA goes to the warehouse or the factory that makes all the proteins, which is the rough endoplasmic reticulum. The UPS picks up those products, repackages and modifies them, and then ships them to wherever they need to go. So it's kind of a cool process. Everything starts from your DNA, and we're going to talk about each of these steps. We'll talk about how your DNA, with the different structures in here, the nucleotides. We'll talk about how it curls up around little proteins called histones. We'll talk about how it winds up in this really tight structure called a chromosome, and how it all fits nicely inside of your nucleus. All right. Here are the major functions you have to know. First, it stores the genetic material. The genetic material is called DNA. Okay? Deoxyribonucleic acid. It stores all of your genes. Next, it controls expression for the genetic code. So not only does it store it, it's like a librarian that says when that information can be accessed, what it can do. So if I want a copy of the blueprint, I have to ask the nucleus for it. The nucleus takes the DNA, the original blueprint, and it transcribes it. It writes it into a recipe for me to take with me, an RNA. So I can't check library books out of here. I can't check the DNA out of the nucleus. If I want to take anything out of the nucleus, I have to take a copy of it. A little short fragment copy of it's called an RNA. That RNA is moved out, and then the RNA is the recipe to make whatever protein I want. Right? So it controls transmission of the genetic information, and this is talking about passing it down. So not only does it control where that information goes inside the cell, but when the cell goes to replicate, it controls the replication process too. Right? And we'll talk about mitosis and meiosis in other videos. Actually, we'll talk about when we start talking about tissues and replicating the cell. Right? Further nucleus anatomy, you need to know these parts of the nuclear envelope, which wraps around the nucleus. So here's an electron microscope picture of the nucleus again. The nuclear envelope is just like the plasma membrane. Same material that wraps all the way around and holds all this DNA inside. Okay? The nuclear pores are little tiny holes that allow RNA to slip back and forth. So RNA can be made in here. I can make a copy of that recipe and slide it out through a nuclear pore. This nuclear envelope is double layered. It's super secure. It's like when we talk about the lipid bilayer, it's 2 layers of these phospholipids. Well, we have a double bilayer, which means we actually have 4 layers of phospholipids in the nuclear envelope. Right? So things getting in and out, it's not easy to do. You have to have these pores to allow them to pass. We'll talk about this nucleolus. So you can see all this DNA is all open up in here. Well, it's kind of wound, but it's all spread out. It's not in chromosomes technically right now. So over in this area, the nucleolus is where it's active. It's where the DNA is being written and copied into RNA. It's basically like if this is all the storage for the library books, here's that center where everybody gets their books together and starts reading at the tables and stuff. That's what's happening here. You're actually making copies. It's the copy center. It makes copies of all the DNA into RNA. Right? And then we'll talk about the chromosomes and chromatin and what's the difference. Right? So the nuclear envelope, I've already kind of jumped the gun on this and told you. It's a double lipid bilayer. It's 2 parallel membranes with a little tiny fluid-filled space in between. So here you can see a phospholipid bilayer. Here you can see another phospholipid bilayer. And then you have all the DNA, the chromatin, and chromosomes inside. Right? There are your nuclear pores. And like I said before, the outer membrane of this is continuous with the rough endoplasmic reticulum. Right? The nucleolus, like I said, this is where the DNA is actively being copied and written. So I would think of it like one of these newspaper plants. You bring all of your information over here, you feed it in the machine, and then it goes crazy making tons and tons of copies. It's printing off all these copies. So the nucleolus just means little nucleus, and it's where the chromosomes are active, or I should say the chromatin, which is the material that holds DNA, is active. So it's the unwound open DNA and RNA and the enzyme machinery that's putting it all together. Right? When we talked about ribosomes are made with the RNA and proteins, right? So the ribosome subunit assembly is actually here, and then we ship them out through the nuclear pores to get out here to actually make proteins. Right? So looking at all these structures again, if we looked on the electron microscope and just kept zooming in, here we can see the whole nucleus. The whole nucleus looks kind of like a little moon. Right? All those little craters in the moon are nuclear pores. That's where RNA slips back and forth. And if we zoom in even closer and look between the nuclear pores, look at that membrane. That membrane, remember, it's a phospholipid membrane, but look at all these proteins that are woven almost like a sheet through there, or a net in there. And they call it the lamina. And it's just proteins. It's taking the nuclear envelope and making it really, really secure and really tight, because whatever's inside of there is really important. If you destroy that, if something gets through and destroys your DNA, that DNA is gone forever. It's like taking a page out of a precious book that you only have one or 2 copies of and burning the page completely. It's gone. So whatever's in the nucleus has to be protected really well. Right? That stuff is DNA. The DNA, when you look at it, we refer to it as a double helix because it's this twisted chain. It's almost like a ladder that you twist. What's amazing, the most amazing about the DNA is that it's a code. It's a recipe for everything that your life depends on. Well, your alphabet has 26 letters in it, A through Z, right? You have 26 letters to make all the words in the world. Your DNA, the recipes for life, everything that you need to make from your DNA is written with only 4 letters. So everything, it's like having an entire alphabet of 4 letters to make all the words. All of these letters are in a specific order that they form sentences that describe what's supposed to happen. And that's just amazing. I was reading recently about this technology where they're taking DNA and they're putting information that you would store on a computer in DNA. They're making a biological computer, basically, biological memory. So they can code things, just like binary code. You only need 2 letters, or 2 digits, I guess, 0 and 1. They're doing this with DNA. So we're taking human evolution, or actually not even just human, but all of animal evolution. We're looking at the DNA, or all living evolution, and we're using it for our own benefit. It's just kind of amazing how that works. But these are the four main nucleotides, or the subunits. So they call them nucleotides. You have thymine, which you represent by a T, adenine with an A, cytosine with a C, and guanine with a G. I always remember good cat, GCAT, good CAT. Another reason I remember that is because if you look at this code, on one strand of the ladder, if it's a G, the other strand of the ladder, right where it's matched up, always has to be a C, good cat, right? And then the A and the T, anywhere you see an A, you see a T matched up with it. It's always in that pattern. And when you get more into genetics, you'll learn more about that and the significance of it. All right? So DNA is packed with proteins. Here you have your DNA. So now you have that ladder. Well, we start curling it up, and instead of twisting into a knot, I don't know if you remember old phone cords, but these old phone cords, if they got twisted, they get into this knot. So what DNA does to prevent the knot is that it will put proteins in here, these things called histones, and it winds the DNA around it when it needs to store it. So as it's winding this up, then it'll wind again, what we call supercoiling. Imagine a rubber band. As you twist the rubber band, it gets really tight. Well, then it will start folding over itself until it gets tighter and tighter and tighter and condenses into this big ball. That's called supercoiling, right? So when we take the DNA and the proteins, this long strip all the way through is actually called chromatin. That's the name of the material now that has DNA and proteins. Each little segment of this we call a nucleosome, right? So here's a little segment, nucleosome. There's another nucleosome and another nucleosome. This whole strand is called chromatin. Right? When we take the chromatin and we make it active and we open up a region of it, we call that active region extended chromatin. That's what you see in the nucleus, is where it's active and being used. It's taking the DNA and making copies of the DNA into RNA so that we can use it. That process is called transcription. Right? Condensed chromatin is when you wind it up and you store it. If we tightly coil this up and it starts forming this like X-shaped looking structure, this is actually called a chromosome. This is all inactive chromatin. It's stored. It's getting ready to actually be ripped apart and put into a new cell. Right? Those chromosomes are the highest level of organization. If we take a cell that's ready to replicate, we let it start stretching out and getting ready to replicate, and then we freeze it and we shatter it. We break it apart. We break its nucleus apart, and we spread out all the chromosomes. A human has 46 of these chromosomes. Right? It actually has 23 pairs of them. So if you go through and you separate these out into size, you'd see the longest ones are paired up, and the next longest, and the next longest, and they just keep pairing them up all the way down, and you'll find that there are 23 pairs. So here again, you can see the structure of a chromosome. It's coiled up chromatin, and then you stick it together to centromere, which is just some microtubules that tie them or connect them together. Right? And then you have a chromosome. When you're ready to split these, when you start studying mitosis, which was one of the next sections we'll talk about, you'll break this right here at the centromere, and you'll separate these things called chromatids. So half of this chromosome is called a chromatid. The first 22 chromosomes are called autosomes. Right? The last ones are special. They're called sex chromosomes. And if you're a boy, you get an X chromosome and a Y. If you're a girl, you get an X and an X. It's the Y chromosome that actually makes you male. What's really cool, and this is new information, is that we're studying something called epigenetics, which we've learned that your DNA, your genes, code for what you're going to be in your life. They tell you genetically what you're going to be, but what's kind of cool is you can change that. You can modify it. You still have the same genes that tell you what you're going to be, but you can change how active they are, and that's called epigenetics. So we can turn some of those genes off in our life when we don't use them. We can stop them. If we need certain functions in our lifetime, we can turn them back on. So it's really kind of cool, and if you ever get a chance to look at it, epigenetics is a study of how DNA works after it's been given to you, after you're growing and changing. All right, let's wrap this all up by talking about how cells age. And aging is actually a complex process. It has lots of processes in it. One theory of aging is free radical theory. You make these things called free radicals, and they're toxic, they're dangerous. They go around and they affect your organelles and they start damaging them. Well, when they damage so much that they can't be repaired, then that cell is going to die. It starts aging and then dies. Right? Free radicals are made all the time. When you're exercising, you're making free radicals. Okay? When you're breathing and living, just staying alive, you're making free radicals. Normally we can neutralize them with something called an antioxidant, but we can't always do that. Right? And then the mitochondrial theory. And mitochondria says that mitochondria, as they age, they just weaken and they wear out.